Hello friends, welcome to the Biological Life Sciences channel. Today in this video we are going to see differences between cloning and expressional vectors, and then we are going to see bacteriophage vectors. In that there are two phage that we are going to study. First is Lambda phage vector, that is phage of E. coli, and then in that there are two types, that is Lambda insertional vector. Two examples we are going to see, Lambda GT10 and Lambda GT11, and then we are going to see Lambda replacement vector, that is EMBL3 and EMBL4, and after that Lambda vector we are going to see M13 Phage vector also. Okay, so let's begin. We'll start with the first cloning vector and the differences between them. So we'll start with cloning vector first. So as the name suggests here, this vector is used for cloning experiment only. Okay, so earlier what happened, before the invention of PCR, the fragments or the foreign DNA fragments couldn't be replicated or amplified. Okay, so here in that case we used cloning vector. So cloning vector is a vector in which this vector can be stably replicated into the host. Okay, so what happens, since it can stably replicate into the host, so we can use this vector and put our fragment of interest into this foreign DNA into this, and this foreign DNA will keep on replicating in this vector when this vector is put into an host. Okay, so this is having all the stable machinery required for replication. So the cloning vector that we can use here is from viruses, it can be from higher organisms, and it can be from bacteria also, that is plasmid. Okay, the main thing to note here is the cloning vectors is only used for amplification of our fragment of interest or its replication or increasing the number. Okay, so it is easy to, and the most important property is it is easy to insert or isolate. So it is generally used for stably replicate or amplify our fragment of interest inside the host organism. Okay, so there will be no expression, so that is called as a cloning vector. So now let's see what is the expressional vector here. Expressional vector, on the other hand, as the name suggests, will be used for expressing a particular fragment. Expressional vectors. Okay, so generally this will, when we are putting our target DNA in this, target DNA means it can be a gene. Okay, so that will be expressed inside the host cell when we put this this expressional vector in the host. Okay, so let's take an example of insulin gene here. So along with the target gene, that is insulin, that is foreign DNA, there has to be presence of number of genes along with that. So this is generally designed for expression. So the number of extra genes that are required here is, first of all, it is enhancer, then promoter element, then there is a presence of translation signal, ribosomal binding site, that is Shine-Dalganos sequence or Kozak sequence. In Shine-Dalganos in case of prokaryotes and Kozak in eukaryotes. Okay, so again there should be a start signal. Start signal means initiation codon, then termination signal. So all these things must be present along with the target gene. So what will happen, this helps in expression of our product. Okay, so what will happen, as this target gene is put along with this all this genetic sequence inside the expressional vector, what will happen? Our expressional vector will express our product, that is recombinant product, into the host cell. Okay. Now there is one problem: how we can select the recombinant product. Okay. So that recombinant product again can be selected or purified using number of strategies. There are popular strategies that we are going to we are going to discuss here. The first way of purifying is attaching histidine tag. Okay, it is also called as his tag. Okay, so these are generally 6 amino acids of histidine that is attached with the gene, that is target gene, along with the other sequences also. So what will happen? Our target gene will be expressed along with this histidine tag. Okay, so now when we are going to purify the insulin, recombinant insulin, the example that we are taking here, so what will happen? This histidine is having a high affinity towards the heavy metals such as nickel. Okay, so nickel is attached in the column matrix. Okay, there is presence of nickel. So our polypeptide, our gene, that is insulin, will be produced in the polypeptidic form, and it will have histidine with it. Okay, so this histidine will bind to this nickel. Now we can easily isolate, or we can easily remove or elute out other proteins by using buffer, specific buffer of certain pH. Okay, and then this histidine can be eluted out afterwards using a buffer of different strength, and then the insulin can be released from this histidine tag. Okay, so this is one way of using the histidine to purify our gene of interest. Okay, the second way is protein fusion partner. So our gene of interest, that can be anything. Here we are taking the example of insulin, that can be fused with different genes. The generally used genes here is GST, that is glutathione S transferase. It is fused along with GST, and it is also fused with another protein, that is maltose binding protein. Okay, so the GST tag is 220 amino acid tag. This also has a particular affinity in chromatography, to which it can be binded to ligand along with the fused protein and thereby eluted after using certain buffers. Okay, this maltose binding protein has a specific solubility in certain solutions, so it helps in increasing solubility of recombinant protein. So what will happen? Our recombinant protein will get solubilized in certain solution, and then it can be eluted out. Okay, so the third strategy that is used here is reporter gene. The generally used reporter gene is green fluorescent protein. Okay, so GFP gene. So what will happen? Our protein will be fused or will be added with the GFP gene, that is green fluorescent protein gene. So what will happen? Wherever this gene is present, our insulin or our recombinant protein will be present. It will show fluorescence and it can be easily isolated. So generally the most common expressional vector that is used in genetic experiments is E. coli, as the regeneration timing for E. coli is just 20 minutes and all the experiments can be easily performed in E. coli. Okay, so this was all about the expressional vectors, the difference between cloning and expressional vectors. Okay, the next that we are going to see is phages, that is bacteriophages. Okay, so what is a bacteriophage? So it is generally the virus that infects bacteria. Okay, so now here it will be like this. The structure is like this. It is having a protein capsid head, middle part, and there is a tail like this. So this is a head, made up of protein, and there is a presence of the genetic material in this, and there is a middle part, and there is tail. These fibers, tail fibers, is helped to attach at the host cell membrane, and the genetic material is injected through the tail fiber, leaving the head behind. Okay, so this is the way of infection of bacteriophage that infects the bacteria. Okay, it can form two types of cycle, that is lytic and so it is based on genes such as CL or CLA gene and CRO genes. Okay, that is responsible for lysogenic cycle, and CLA gene that is responsible for lytic cycle. Okay, so if it goes for lytic cycle, then what will happen? The phage particle will be synthesized in more amount, and this genetic material will be injected into the host. The viral particle will be synthesized inside the host using that genetic material, and that genetic material will be packed into the viral particle inside the bacteriophage particle inside the bacteria, and it will increase in number. So one point will come when that bacteria will burst, releasing the phages. What will happen in lysogenic cycle? The bacterial DNA will integrate into the chromosomal DNA, thereby replicating itself, and it will also form its number of fragments. Okay, so now this number of fragments will be bulged out from the cytoplasm of that bacteria, and the cell here will act as a factory for synthesis of that phage. Okay, so these are the two ways through which this bacteriophage infects. So the two bacteriophages that we are going to see here is first Lambda phage, and second is M13 phage, and how they are used in cloning. Okay, so the vectors which is prepared from Lambda phage are of two types, that is Lambda insertional and Lambda replacement. Okay, so let's see this one by one. Okay, we'll begin with the Lambda phage vector. Let's see what is the Lambda phage and its details. Okay, so Lambda phage, it can clone large fragments of DNA. So generally the size of the genome is 48.514 kb. Okay, so it's having a large genome. Around there are 50 genes present in this that are required for the infection of Lambda phage. Okay, so half of this is essential and half is non-essential part, but that is present in the Lambda phage genome. Okay, so and the most important character of this is presence of cos site. Okay, so the cos site is like this. Suppose this is the whole genome of the Lambda phage, and there will be presence of a site like this on both ends. Okay, so what will happen? This site is called cos site. The full form of this cos site is cohesive site. Or cos. Okay, so what will happen? This cos site is present on the both ends of the genome of Lambda phage, and this helps in circularization of this phage genome when it infects the bacteria. So it is single stranded DNA, that is Lambda phage genome is single stranded DNA, and it will get circularized using this cos site. Okay, so this cos site is very important in cloning experiments of infection. First of all, what will happen? Let's see the replication of this Lambda phage and infection, how it infects. So first of all, there will suppose this is the bacterial cell. The phage is like this. Okay, it's having a single stranded DNA. Okay, so this single stranded DNA first of all will be injected. There is a presence of cos site on both ends. Okay, so using this cos site, first of all this single stranded DNA will be converted into double stranded form, that is replicative form. Means in this form the genome of phage can replicate easily. Okay, so now this replicative form will increase its number by replicating it is replicating using rolling circle model. Okay, so now the replicative form which is formed here will be again converted into single stranded form along with the cos site, and the presence of, depending on the conditions and presence of the gene, either it will bulge out from the membrane along with the phage, or it will form a viral particle inside the bacterial cell. Okay, so when it forms the viral particle inside the bacterial cell, what will happen? This particle inside the bacteria will increase, the number of this particle will increase, and at one point this cell will burst away and these particles will be released. So in lysogenic cycle, the viral particle will be bulging out from the cell membrane of the bacteria, and the bacteria will not die, but it will be like a factory of production of these viruses. Okay, so this is the way of infection or replication cycle of a Lambda phage that we saw in details. Okay, so now let's see what are the different vectors that are prepared from Lambda phage. The first that here we are going to see, one thing I forgot to mention, the 23 kb to 25 kb of fragment can be cloned using Lambda phage. Okay, so first that we are going to see here is Lambda insertional vector. Okay, so as I told you that Lambda phage is having half of the coding sequence that is required for infection and half of the genome is non-essential one. So we can insert our fragment of interest here into the insertional vector here. There is, the size can be about 35 kb of the DNA, and here we can insert about 18 kb of foreign DNA into the phage particle. Okay, so this much amount maximum is used as a coding sequence which is required for infection, and we can clone around about 18 kb of insertional vector. The first insertional vector that we are going to see here is Lambda GT10 vector. Okay, so let's see what is Lambda GT10. Here it is used in cDNA cloning in bacteria. cDNA is prepared from mRNA from eukaryotic cells. So now this cDNA will be cloned in bacteria using Lambda GT10 vector. The selectable marker gene here used is CL gene that produces a repressor for lytic cycle. So what will happen when CL gene is producing repressor? It will produce or the cell will undergo lytic cycle. Okay, so repressor for lytic cycle gene, so it will repress the lytic cycle gene. Okay, so what we are doing here, there is a presence of multiple cloning site in the CL gene. Okay, so what will happen, the foreign DNA fragment is inserted into the CL gene. Okay, so it will become CL negative. Means what? The CL gene is insertionally inactivated. So now it is not going to produce repressor that is required for lytic cycle, and the cell will go for lytic cycle. Okay, and it will form a clear plaque. Means what? The recombinant cell will form a clear plaque, and the non-recombinant cell will form a cloudy plaque, as the gene CL is intact in that, and it will go for lysogenic cycle and it will form a cloudy plaque. So the clear plaque of the cells we can select for the presence of our fragment. So this was the cloning experiment that is done by the insertional vector Lambda GT10. Okay, so let's see the next insertional vector, that is Lambda GT11. Okay, so the only difference here is here we are using the beta beta-galactosidase gene, that is LAC-Z gene. Okay, the size here is around 43.7 kb, and here also we are using it for cloning of cDNA, that is cloning of cDNA using the lambda GT10 vector. Okay, so what is happening here? The multiple cloning sites is present inside the beta-galactosidase gene. The multiple cloning site is present inside the beta-galactosidase gene. So what will happen? This beta-galactosidase gene is insertionally inactivated. So when this is insertionally inactivated, the gene will be the gene product, that is beta-galactosidase enzyme, will not be formed. Okay, so what will happen? We use a substrate here called XGAL for the bacteria here. So what will happen? The genes or the bacteria in which the vector is having beta-galactosidase intact will be able to degrade this XGAL and produce blue color, while which is having our fragment of interest, that is foreign DNA, will be producing white colored plaque as the virus is infecting the bacteria. Okay, so this white colored plaque can be selected and we can go for isolation of our fragment of interest. So these were the two vectors of lambda phages that is used for cloning experiment. So now let's see the next vector, that is M13 Phage. M13 Phage. So this is also a bacteriophage that infects E. coli, but the peculiarity here is it only infects the F plasmid or sex pillars containing cells. Okay, so what is the genome here? It is single stranded circular, it's single stranded linear DNA. The size of the genome is around 6,407 base pairs, out of which we can clone around 500 base pairs of foreign DNA. So comparatively with the lambda phage, the foreign DNA that can be cloned here is around 500 base pair. Okay, and this phage lands on the sex pillars of the bacteria and infects that bacteria. Let's see this infection cycle. Suppose this is the bacteria. Here there is a presence of sex pillars on both sides, and suppose this is the filamentous M13 phage. It is having a single stranded DNA. Okay, so now this will go and infect the sex pillars. It will insert its single stranded circular DNA inside this. Upon insertion, since there is a presence of cos site here also, there is it will go and circularize into the bacteria. Okay, so now this is the replicative form. It is also called as theta form. Now this is replicated into number of copies using rolling circle model. Okay, so after that it will form again the single stranded copies. Okay, and this will be packed into the sex pillars again, and this M13 phage will be bulged out from the cell along with the cosmid on both ends, and thereby increasing its number. So this is also having, so this is the way by which this M13 phage infects the bacteria. So now here what will happen, the essential part is kept in the bacteria intact, that is essential genome, and the non-essential part is added with the foreign DNA of interest. We know that the 500 base pair around fragment can be inserted into this. There are few advantages of using M13 phage. The first and foremost advantage is the double stranded DNA which is formed inside the bacterial cell, that is the replicative form, can be used as a plasmid. It looks like plasmid, and since it is in bacteria, so it can be used as a plasmid, that is double-stranded replicative form. The next is the single-stranded DNA of this phage can be used as a template for sequencing. Since this is single-stranded only, and in sequencing single-stranded phage is used, so if the phage is having a single-stranded template along with the fragment of interest, that is foreign DNA, it can be easily taken for sequencing directly from the phage after infection. So this was M13 Phage that is used for cloning. Okay, thank you so much. If you like the video, please press like button, share, comment and subscribe. Thank you for watching.